identification and characterization of the small molecules which can activate Wnt beta-catenin pathway, which potentially can regenerate hair. So just Professor Gonzalez already told you about this, so I don't want to tell you furthermore. Anyhow, this Wnt pathway potentially can be used for development of drugs which can regenerate hairs. So what we are doing, actually I'm not a dermatologist. We do signaling pathway with various pathophysiological roles of this pathway. So anyhow, this pathway is one of the pathophysiological roles related with hair formation and birth. Anyhow, I would like to briefly tell you about this beta-catenin pathway that make you more clear. Anyhow, without Wnt stimulus, this key signaling molecule what I'm going to tell you about today, beta-catenin, form complex of the several of the negative regulators of the Wnt beta-catenin pathway, including APC, Xn, and GSK3 beta. By forming this destruction complex, beta-catenin subject to the phosphorylation and terminus and followed by ubiquitin-dependent proteasomal degradation. With Wnt stimulus, this destruction complex is now dissociated and beta-catenin is stabilized and translocated into the nucleus for the activation of target genes. Therefore, amount of the beta-catenin and the nuclear localization of the beta-catenin is important to indicate for the signaling. So far around 100 target genes are identified. Among them is several is important for the hair formation and wound healing so on. As our initial study for a while ago, we screened the drugs, compound libraries which can activate with beta-catenin pathway. We identified such kind of compound. However, we do second screening. This second screening is very meaningful. We used neuronal stem cell, which primary cell. When we treated compound, some of the toxic compound easily actually killing these fragile cells. Therefore, we do second screening, good candidate we characterized, and in vivo studies we perform, including this kind of hair studies. Actually, first identification of this kind of compound is valproic acid shown in here, which is used for the epilepsy or something like a mental disease. So what we for several decades, what we identified is valproic acid uniquely activate beta-catenin and induce the hair forming. Especially in the anagenic phase, you can see the equivalent to the valproic acid. It formed hair very well. Alkaline phosphatase stain is shown in here. Activation of Wnt in the hair follicle, we used Takal mice activation shown in here. So interestingly, we see in the ALP is strongly induced by valproic acid, and the BMP4 and the 6 BMP4 is a target of the Wnt pathway is activated. So what we noticed the difference between the valproic acid and the minoxidil. Similar thing is what we noticed the AKT and ERK activity is increased. There is similar to the minoxidil. There is a hair forming activity. But however, what we see the uniquely, this valproic acid actually stimulate, activate beta-catenin and induced activated stem cell markers, keratin 15 and CD34. Also by wound generation, what we see in here, this is the hair forming, which is observed by Professor Gonzalez. So we identified several of the compound and we further progress analog synthesis happening for this compound. So I don't want to mention in detail. Right now I am telling you about the novel thing. After we identify valproic acid and the small molecules, what we observed, there is some kind of problem. This results in the hair formation, even human, maybe. However, there is some kind of negative feedback is a function. Therefore, the hair size thickness is actually low, and also there is some hair formation is limited by that kind of thing. So for a while ago, we investigated this IDBF. We named it. What is IDBF? Which is actually identified one of the similar protein with IDBF, IDATs. IDAX is first identified by Professor Kikuchi. He is currently at Osaka University. So that is actually binds to the disabled and function as a negative role. So this kind of disabled binding domain was well conserved for our proteins. So actually we thought this is negative regulator. So we confirmed that, but later we identified uniquely this factor is induced. So induced by Wnt signaling, then it functions as a negative feedback regulator. So signal-dependent induction plus actually signal-dependent disabled interaction actually result in the negative feedback role. So what we see that kind of phenomena more clearly when we see the hair cycle in mice. Beta-Catenin is actually androgenic phase significantly increased, but we observed IDBF is also induced. But if you see the carefully, the induction time, activation time for the beta-catenin shown in here, and the IDBF is different. So a little bit later. So what we know that after Wnt signaling, this IDBF is not a direct target. However, it transcriptionally induced that function as a negative feedback role. So when we see that this hair follicle lesion, keratin 15 and the IDBF, that is totally opposite in this kind of ORS region. We actually tested the negative role of the IDBF by using human dermal papilla cell. When you overexpress IDBF, what you can see here, beta-catenin and ALP is reduced. So similarly, you can see the transcriptional level, alkaline phosphatase staining. We confirmed that by cytochemistry studies. When we knock down, oppositely, when we knock down IDBF, then increased beta-catenin, ALP staining is increased. So this negative regulator, IDBF, which is we named because this is inhibitor of disabled and bone formation. That's what originally we see this phenotype. When we made this knockout mice, but we see the phenotype, one of the role of the Beta-Catenin pathway, the bone is longitudinal growth of bone is hugely increased, bone mineral density is increased, which is unknown, unpublished yet. However, we see that this mice is very interesting thing is here, adipocyte lineage is highly increased in here, hair follicle is further developed. So adipocyte is increased is a little bit confusing first. This morning I told with Professor Kotsaleris. However, later I knew that one of the speakers in the plenary lecture today mentioned about that. So So this Wnt beta-catenin pathway is sometimes activated. Originally, it activated adipocyte differentiation is inhibited. But in this specific case, it's a thermal system. When this Wnt pathway is activated, some adipogenesis is increased. That's what we see this result. This morning I really appreciate for that kind of information for me. Anyhow, this kind of result make me knockout mice actually increase Wnt beta-catenin pathway activated, that increased the hair formation, hair follicle is increased. One interesting is we hypothesize if the Wnt pathway is activated, then negative feedback is blocked, then we can expect this kind of huge hair follicle growth. So what we see in here, relative dermal thickness and the relative number of the hair follicles is hugely increased by knockout mice together with the valproic acid treatment. So you can see here, in this case specifically, keratin 15 and the CD34 stem cell marker is critically increased together with the beta-catenin induction and other these markers. So we further characterize this immunoblotting, and this is the histochemistry. You can clearly see with this knockout together with the valproic acid treatment, you can see the hugely increase in the hair follicle markers. Also stem cell markers, including LGR5, is also strongly induced. That kind of result is further correlated with this kind of hair formation, even the adipocyte lineage formation in here. So Then we just really wanted to know that hair regeneration and hair follicle neogenesis. But you can see in here we use uniquely, in this case, 7 weeks mice. That is different with Professor Cosaleris used 3 weeks or like that. Also, we used 25 days in here. What that means by a little bit aged mice we used. Also, we want to use the short time whether this kind of hair follicle neogenesis is happening. This wound introduced in here, hair is formed, but this hair regrows, anyhow, some kind of effect of the cell. However, you can see in here, white hair formation like a situation. This knockout together with valproic acid treatment, sometimes it can induce hair neogenesis. So this is, we think, is critical. And keratin 17 was highly induced in this case. Finally, what I'm going to tell you about, we just wanted to know whether this kind of phenomena, we can see any correlationship with the humans. So in this case, actually, we got several of the this bald specimen, and what you can see the female non-bald and female bald. Also, only I showed a very limited case in here, but we do several of them. So what you can see in here, non-bald, what you can see beta-catenin is critically increased. Here is IDBF maintained very low level. That's what we normally see in here. Stem cell markers is somehow activated in these hair cells. So what you see in here, female bald case, here is the hair follicle is most of the miniature lost. So IDBF is interestingly highly increased in here. Oppositely, beta-catenin is maintained very low. So what we think this kind of high, I don't know the reason, but high level of the IDBF could be some kind of reason, weak activation of the beta-catenin, that could be a potential some relationship with the bald, whatever. So we also staining by DAB staining, what you can see in here. So beta-catenin is highly induced in this hair follicle in normal case, again. So IDBF maintained low level. However, female bald, you can see the beta-catenin is maintained very low, IDBF increased high. I'm not showing individual data for human case, so several of the cases we tested, but if you take my message, we could observe a similar kind of phenomena. Okay, finally, what I'm going to tell you, we are doing various pathophysiologies related with Wnt pathway. But today, what I told you all about these molecules or some kind of mechanism related with this hair is Sung-Hoon Lee is actually, he is the graduate student, the senior graduate student. All this hair forming or wound healing, what we are currently doing, he performed in this meeting, he participated. Hyun Lee is the make knockout mice around something like 7 years ago, and we continually use that mice and other phenotypes too. Finally, thank you for your attention.
Now open for discussion.
Can you tell us a little bit more about the compound you're using? How did you find it?
So what you say is negative feedback, or just a compound which activated with beta-catenin pathway? Which one?
The screening. Yeah.
Screening is actually we are okay. So I'm sorry I did not mention in detail, but HEK293 cell which have a chromosomally incorporated topopolation reporter. The reporter have 7 TCF per beta-catenin site. Therefore, the reporter is very sensitive, and that's what we used for the initial screening. Then we used more physiological cells used for the test of those kind of phenomena that we do currently. However, if I emphasize one more time, we use a secondary screening. We think very important because most of the toxicity of the compound, some of the problem, we can initially reject out those compounds. Therefore, we bring those good compounds for our future studies as we are doing.
Yes, please.
I have one question. So you mentioned about the IDBF. Yeah. So you showed some figure about different formation compared to white type. So is there any other kind of side effect or any phenotype when you knock out IDBF?
Okay, so actually I'm just focusing on the talk related with hair formation in here. So knockout mice induce the hair formation, but as I told you, we actually generated this knockout mice about 7 years ago. However, this gene knockout mice is so far was not published yet. So what we do with lots of phenotypes observed. As I already showed, this knockout mice phenotype is bone mineral density is critically increased, and also longitudinal growth of bone is increased. Therefore, we use this as a target for anti-osteoporosis drug development too. And also, these mice have a very interesting phenotype. Normally, this mouse behavior is the same as normal. However, what happens if we give a stress to these mice? Later, these mice became very, very aggressive. So it is easily attacked to even the large animal it can attack. But normally, okay, no behavior change. However, when the stressed, so isolation, such as long time isolation, result in stress, then very aggressive. And other phenotype is also shown, and I want to hit some of them related with kidney development or so on.
Hello, thank you for your nice lecture. My name is Myung from Yonsei University. I have just one question. If you wanted to know about the IDBF, why did you use a female bald specimen rather than a male bald specimen? Because female bald specimen, it is very, you cannot tell if that is a bald or not.
Okay, so I just expect that kind of question. Only thing is I show the case, what we have it. But I'm sorry, I did not mention, I'm very sorry. The professor wants to provide this hair specimen. So we have several of the male and one female, that's what we do. But if you take my message, male case was a similar phenotype. So CXX5, that is actually IDBF, that is highly induced. And however, Beta-Catenin is reduced in case of bald compared with.
Are you saying that the male had similar?
Similar,
but
yes.
not, you didn't
Yes,
touch
yeah.
Okay, thank you.
Yeah.
And let me ask, how many patient sample you?
Yeah, only we have we got 4. So one female, the other is actually. So we do systematically many of the human cases. If we get, we will further progress that statistically. But very interesting why this CXX by IDBF is such a high level in birds. That's the question. So if that is true, our target is wonderful. If I give a message, we are actually developing, currently working with blocking of this negative feedback by using peptide and a small molecule. That's what we are currently on the way.
And one more question, sorry. The biphoric acid treatment, how long did you treat the compound?
It depends on what kind of situation, but we do something like, I already showed some of the 25 days or whatever. So it depends on our purpose. So as I already told you, neogenesis and or some of the normal case, whatever, we can vary. So I cannot say exactly.
So you didn't try longer than 25 days?
No, we do long time. So eventually, such as if we make a wound, 80 days, if we grow, we can definitely see lots of white hairs that is newly grow hair.
Okay, thank you.
Okay, thank you very much.
Presentation.